the questions I get frequently asked these days, we are in August 2021 these days, is if I'm tested positive for COVID, how do I know if it's a Delta variant or not? I was asked this question by many patients, in fact, many staff members, including nurses. So I thought I'll share some insight into this and try to clarify this doubt. The simple answer is whether or not you had a Delta variant COVID infection, the simple answer is no. You will not know whether it's a Delta variant that you had or not, because the tests we do here will only tell us if it's positive or negative, if it's a COVID infection or not. It will not. The only way to know what type of variant it is, whether it's a Delta or whatever variant, is to do a genomic analysis. Normal labs that we have outside that gives the test results very quickly will not do genomic analysis, because genomic analysis involves much more resources. It takes a lot of time, in fact many days sometimes, to do a genomic analysis, and it is only done by certain organizations such as CDC, health departments, state health departments, certain research labs, genomic labs, universities like Johns Hopkins University, public health institutes. This is done in specialized labs where they have to do genomic analysis. Let me try and explain what a genomic analysis is in reference to this question. This genome, which is the inheritable material of the cell, we know DNA and RNA, and you might have studied in high school or in college about nucleotides, right? If you don't worry about it. I'll try and explain it in simple terms. Nucleotide is the simplest building block of a DNA or RNA. You must have heard terms like AC, GC, ATGC. These are the nomenclature, the names given to these nucleotides. The simplest building block of a genomic material is called a nucleotide. In the case of the COVID virus, there are 30,000 such nucleotides. It's an RNA molecule, it's not DNA, it's an RNA molecule. This RNA molecule has 30,000 building blocks or nucleotides. So every time this COVID virus, this coronavirus replicates, which means divides and multiplies and in the human body, this 30,000 nucleotides replicates and forms a new virus, right? And generates a new virus. In this process, sometimes errors can happen. Mistakes in the replication can happen. So when these mistakes happen, a new variant may be produced. This new variant virus can be stronger, can be weaker, or it may not show much change, depending on how these mutations happen. So some variants are stronger, they survive and become more virulent. Virulent meaning they become stronger and have the capacity to survive longer, to multiply faster, and to spread faster and cause severe infections. So this Delta variant is one such variant. When the original virus mutated, or when the original virus replicated, these 30,000 nucleotides, some of them got mutated and formed a new variant which became more virulent, has the capacity to cause more damage to human body. So these changes in the nucleotides can only be detected by genomic sequencing. You cannot detect them by a normal lab test that tells you a positive or negative. No, if you are positive of COVID, then the health departments, research institutes, genomic labs, they take this positive sample and run a genomic sequence on it. And that's how they have done it. They have to, in a state in a section of population of all the positive people that they have collected the material from, and they have run the tests. As of August of 2021, up to 84% of all positive COVID were tested to be Delta variants. So if you are positive for COVID infection, it's highly likely that it is a Delta variant. So they're assuming that almost all or most of the cases of COVID positives are Delta variants, and therefore, that is the most dominant variant in the world right now, and most commonly in the United States. This Delta variant, as we have seen in old videos, was detected first in India, in the state of Maharashtra, in October 2020, and since then it has spread to the rest of the world. So the quick answer to the question, how do I know if my variant, if I'm tested positive for coronavirus, if that is a Delta variant or not? The simple answer is you will not know it unless you run a genomic sequence, and genomic sequence is done only by selected labs, certain institutes, research labs, health departments, and only they can tell. So if you're in a hospital, if you're admitted, you're unvaccinated, and you got a severe infection, assume that you have a Delta virus, Delta COVID variant.